To process information, cells need the right tools for the right job. Let's look at case study number 2, where we determine what part of the gene sequence actually is the promoter portion. We'll first look at the experimental design, where the investigators took a gene, a double-stranded piece of DNA, the full-length gene, and mixed with that the RNA polymerase. The RNA polymerase would bind to the double-stranded DNA, and the investigators would then add DNase. DNase would chew up any exposed DNA not covered by RNA polymerase. Once they had chewed up the non-bound DNA, then they could see the sequence that was bound by and protected by the RNA polymerase. They took this sequence from a particular gene in the virus called T7. It's double-stranded DNA, and it's undamaged because the RNA polymerase protected it from the DNase. They also could deduce from there which portion of the DNA encoded the mRNA, and it extends off to the right where the start codon is further downstream from this promoter region. They took the sequence from virus T7 and compared it to 3 other viral sequences of promoters, as well as 2 bacterial promoters, including the lac promoter that we just studied in the previous case study. Inside the red box are conserved nucleotides, and you can see there's a whole column of T's here and there, and a column of A's. So by eye, the investigator was able to figure out which nucleotides formed a consensus sequence for a promoter. They also coined the term or the usage of a +1 indicating which nucleotide was the first nucleotide transcribed downstream of the promoter region. If you did integrating question number 1, you looked up a human gene sequence and you found that even some human genes have highly conserved promoter sequences similar to the viral and the bacterial promoters that are very rich in the nucleotides T and A. Mathematicians and computer scientists have gotten together to start the field of bioinformatics, and in this particular BioMath Exploration, we learned how a position weight matrix is used and created. And the position weight matrix is a way to determine whether a particular nucleotide sequence will function as a promoter. In this case, we have the promoter region shown up above, while at the same time quantifying the values for each of the letters. So the first nucleotide is T in the weight matrix, and it's T in the promoter. And if you add up these values and it is a value greater than 1, then it likely is a promoter. In this case study, we found out that the promoter is a sequence of DNA nucleotides upstream of where the mRNA is produced at nucleotide +1.